Good evening to everyone. Thank you for attending. I wish I could see your beautiful faces, but I cannot. It's somewhat weird to give a seminar to people that you cannot see, but I'm glad that you're here. So without further ado, what I'd like to do today is to give you a quick seminar. It's going to last maybe 30 minutes, not more than that, on understanding COVID-19 testing. And this is not a specialized course, it's a crash course. One thing I'd like to do is to start with, wait a minute, let me see now why my controls seem to have frozen. I'd like to be able to give you a brief overview of the virus, and then discuss some of the use cases. How do the needs of a medical doctor differ from those of a government official? Then we'll look into the aspect and accuracy of the tests. I'd like to give you a quick note on immunity passports, and finally I'll describe our approach before trying to answer your questions. So let's start with some nomenclature. SARS-CoV-2 is the name of the virus, and COVID-19 is the name of the resulting disease. That's very much like HIV versus AIDS. HIV is the name of the virus, and AIDS is the resulting medical condition. By now, all of you have seen pictures of the SARS-CoV-2 virus. This is the structure. And the virus is made of only 4 different proteins, but multiple replicates of each protein. You have these red spikes here that serve to attach and then invade human cells. And then you have this gray envelope that encapsulates the virus genetic material that's called RNA. And for today's presentation, we'll keep it simple, and we'll just say that viral RNA is very similar to the human DNA. So humans have a sophisticated machinery that I have depicted here with a scene from Charlie Chaplin's Modern Times. And this machinery reads the code embedded in the DNA and synthesizes proteins accordingly. Now, human cells can do that, but viruses do not have this machinery, and they cannot replicate alone. So the virus must hijack the human machinery in order to proliferate. The virus starts by entering a human cell, which is here depicted with this rectangular box, and it injects within the human cell some RNA. Then the human cells are fooled into reading the viral genetic material and into making proteins accordingly, which then can self-assemble to give the virus. So while the virus is doing that, in the meantime, humans are detecting that there's a foreign pathogen and they're fighting back with antibodies. So people will develop antibodies against sorry, give me a second here to minimize one window. It's not working for me. People will develop antibodies against the virus proteins, and these antibodies take 1 to 3 weeks to appear, and then they persist for months with the goal of protecting against future infections. And for those of you who like to crunch numbers, 30% of the people develop antibodies within the first week, so 1 week within the first initial symptoms, and 90% of the people have antibodies after 3 weeks. So from a diagnostic development standpoint, this has the following implication. Diagnostics can be made to detect 3 different things. You can detect the viral RNA, you can detect the viral proteins, or you can detect the human antibodies generated against those viral proteins. If you're detecting viral RNA, it's called a molecular test, or also called a nucleic acid amplification test, an AAT. And PCR is perhaps the most popular type of molecular test. If you're trying to detect the viral protein, it's called an antigen test. And if you're detecting the human antibodies, then it's called an antibody test, or also a serological test. So these are the 3 broad categories of testing. So what are the potential use cases for these different types of tests? The first one is the one of a medical doctor. So a medical doctor wants to know if his or her patient is infected and requires treatment. That's an obvious use case. But perhaps even more importantly, because right now a lot of the patients are just sent home if they're not in if they're not severely sick. But a more important use case is that the doctor will need to manage the patients that are being admitted to the hospital for whatever other reasons. You need to know if those patients are potentially contagious. And so what's needed there is the ability to detect active infections. So you need either to be able to detect RNA, so you need a molecular test, or you need to be able to detect proteins, so you need to detect an antigen test. What's not appropriate is an antibody test, because the antibodies appear late, 1 to 3 weeks after the first symptoms, and because they remain in circulation once the person is healed. Now, at the other extreme of the spectrum is a use case that I think is very interesting, and it's the one of a government official. So a government official, unlike a medical doctor, does not care about individuals, but about populations. And that official needs to be able to make public health recommendations. So here is an example of a question that could float through a government official's mind. For instance, in order to reopen schools, I need to know if children aged 10 to 12 in school district, whatever, have historically been asymptomatic carriers. Well, for this, an antibody-based test is the best option, because the antibodies will give you the history of a population. You may test today 100 students with a molecular test and not find a single case of active infection, but an antibody test may still reveal that whatever percentage of the students have been infected at least at some point in time. So that is very useful data, but it's data that's more useful, again, for public health recommendations than for individual case management. Then a third use case is just you and I. So for instance, I've had symptoms in February. I'm curious, was it really COVID-19? Well, for that, an antibody test would be ideal. The problem is that they're not yet allowed for at-home testing. Or I'm feeling fine, but could I be asymptomatic, and do I pose a risk to my family and colleagues? Well, here it's a molecular test that would be ideal. But again, it's expensive, it's a hassle, especially if you need to do that on, say, a weekly basis. And it needs to be done by trained personnel. It cannot be done at home. So unfortunately, currently, there's just no convenient solutions. There's only cumbersome roundabouts. A fourth use case is the one of an employer. So an employer may have many employees, some of which must meet with customers face-to-face. How do you manage this responsibly? Well, my suggestion would be to do both a molecular test on a frequent basis, maybe by spitting in a tube and sending this to a lab, and in addition, to perform an antibody test, at least initially, to know the baseline situation. The cost of the antibody test is really minimal, but it brings you useful information. Not only you know what the baseline situation is, but you could also garner additional information. There we go. So I believe we were on this slide when things went wrong. And what I was just saying is that regardless of the platform, there are no easy solutions for routine at home or at work testing. So let's have a look at how the different tests look like and what their accuracy are. So molecular tests are often run in clinical labs like this one, using very sophisticated and expensive instrumentation. Molecular tests can be run on this type of clinical analyzers. Here is an example. It's the Roche Cobas 6800. And these instruments have a high throughput, they're very accurate, and they can churn out about 100 results in 3 hours, but that does not include the logistics of sending the samples to the lab and then of sending the results back to the physician and the patient. Now, molecular tests can also be run on benchtop instruments. And here are two examples. One is the GeneXpert from Cepheid, and the other one is the Abbott ID Now device. They both employ a system of cartridges. Here it's a single cartridge. Here it's two cartridges, a blue one and an orange one. And you insert your sample in it, and you can get the result within 15 minutes. So these are very powerful systems. But there's a caveat, and it's that molecular tests are only as good as the sampling method. So the CDC still recommends nasopharyngeal swabs, which remain the gold standard to date. These nasopharyngeal swabs require trained personnel, and they consist in inserting a very thin and flexible swab through the nose, and then you have to go horizontally, go past the delicate structures of the turbinates, and reach the very back of the throat, pretty much at the height of your ears. So this is the nasopharynx. So needless to say that this is not a convenient sampling method. So people are looking for substitute. If you could at least go through the mouth and go through the oral pharyngeal region, that would be an improvement. But saliva would be best, or perhaps something just collected from the tip of the nose. So these people are looking into that, but right now it's not fully demonstrated that these alternative sampling methods are as good as the nasopharyngeal swabs. The antigen tests also require a swab. They also require an instrument, and instead of requiring a cartridge, they require a cassette like this one. The sensitivity is about 85% and the specificity around 100%. And it takes about 15 minutes to read a test. As far as I know, there's only 2 devices that have been authorized by the Food and Drug Administration, and it's one made by Quidel here in town in San Diego, and one made by BD Biosciences. And finally, the antibody tests can be run on anything, from clinical analyzers to the benchtop instruments we just saw, to things that can be just read with the naked eye, like this one. They require blood that's collected 3 weeks after the onset of symptoms. And if they're run on a clinical instrument, then the tests are very sensitive and very specific. The portable tests, like this one, they're convenient and they're cost effective, but the market has been flooded with tests that have poor performance. So the FDA has withdrawn more than half of the tests that it initially authorized, and is compensating by requiring a medium to high complexity lab to review and perform the test. And that really defeats the portability of these tests. I should add that there are ways around it, and you can submit additional data to the FDA to have the test at least authorized to be used at the point of care. But anyways, so right now you cannot use these at home. So in summary, the molecular tests are, and it's difficult for me to put a figure here because the molecular tests are the gold standard. So what are you going to compare them to? So say that the molecular tests are around 98% sensitive and 100% specific. These figures are really hard to define with precision, but what can be said is that there's a very good concordance between different manufacturers. These assays, as I mentioned, do still require a well-performed nasopharyngeal swab. Then there's the antigen test, about 85% sensitive and 100% specific. There's only 2 devices in the US that are authorized. There are the antibody tests that can be run in a clinical lab. And this is interesting. So for instance, some manufacturers claim 98% sensitivity, but when these tests are actually cross-examined by other groups, these other groups find closer to 85% sensitivity. Specificity is pretty high. And finally, there's the rapid tests, the portable ones, where the FDA now demands greater than 90% sensitivity and greater than 95% specificity. And as I mentioned, pulled out 45 of the 77 previously authorized tests. So as an intermediate summary, I like this quote by a gentleman from the University of Washington that was quoted in Science magazine a couple of months ago. What everyone wants is for a test to be cheap, accurate, and fast, but you can only have ever 2 of those at the same time. And it's true, but I would push that even further. I would say that the challenge will be not only to make a test that is cheap, accurate, and fast, but also one that requires a simple sampling method, so not the nasopharyngeal swab, and that can be amenable to routine use at home and on the workplace. So now I'd like to say a few words on immunity passports. So there are a number of potential applications for an antibody test. One is to determine the history of a population, as we mentioned previously. And the second one could be to issue an immunity passport. So that's a tantalizing possibility, but it's unclear if it's a realistic one. And immunology is a very complex science. I don't mean to have ready-made answers for you, but I want to give you at least a sense for some of the questions that are being asked. One is a matter of short-term versus long-lasting immunity. So the bad news is that the concentration of antibodies to SARS-CoV-2 declines by about tenfold after 2 months. This data was just published by a team from King's College London. And here's the graph from their publication. So on the x-axis, you have the days that have elapsed since the beginning of the symptoms. And on the y-axis, you have the concentration of neutralizing antibodies in a logarithmic scale. Each patient is described as a straight line. And if we take this patient here, these were the levels of antibodies about 12 days after infection, and 2 months after infection, they have declined. And that's true for every single patient. Since I have this graph here, I will open another parenthesis. And it's that on this graph, the red lines are patients that were severely ill, so hospitalized. And the black lines are people that were not hospitalized. So you see that people that are in hospitals have much higher antibody titers than the others. And that's probably because they are more sick and the body is trying to fight back as hard as it can. And this may also explain why some of the antibody tests that were initially authorized did not perform as well as expected. So the first tests that arrived from China were tested on people who were recently infected and that had very severe symptoms. So they were evaluated with a population that had high antibody titers. Now, a couple months later, we want stuff that works not just for this group of patients, but also for people who've had mild symptoms and who have had more than 2 weeks elapsing since the beginning of their symptoms. It's a big difference. You're going from titers of 10,000 to about 100. So now the tests need to be about 100 times more sensitive, if you will, than what they were required to be at the beginning. So the bad news, again, is that the concentration of antibodies declines with time. The silver lining is that antibodies are produced by B cells, of which a fraction can become memory B cells. And those memory cells circulate for a decade and can accelerate the immune response if there is a subsequent infection. So the question is, how effective will the memory B cells be in the long term? And also, by the way, do we have memory B cells against other coronaviruses that could afford at least partial protection? So this is about the time course. Another question is one about what I would call regular versus neutralizing antibodies. So normally antibodies bind to the antigen, so to the viral protein, and then they target it for destruction. The antibody-antigen complex gets swallowed up and digested by specialized cells called phagocytes. So that's what normally happens with an antibody. But some antibodies can also bind to the antigen and prevent it from performing its natural function, such as invading and therefore infecting human cells. And these antibodies are called neutralizing antibodies. For instance, we said earlier on that the virus attaches to human cells and invades them by means of this spike protein. And in fact, it does so via a very small domain on this spike protein called RBD, receptor binding domain. And it's likely that antibodies to this receptor binding domain will prevent the virus from entering the cell and infecting the human in the first place. So one of the questions is if these neutralizing antibodies will confer more protection. And another one, by the way, is if one should look for the antibodies in the blood, as most people are doing now, or should we be looking for them in the mucus that's lining the respiratory tract, because this is the first mucosa that comes in contact with the virus. So again, I don't have all the answers, especially since the virus has been around for only 6 months. My words of wisdom, if I can call them like that, is that it may be like the flu. If you get it, you're probably protected in the short term, but perhaps not in the long term. And the CDC on its website states that the antibodies are likely to provide at least short-term protection, but also warns that there is no reliable evidence. All right, so what is our approach? Well, there is a fifth use case that we did not describe before, and it's the one of low-income populations. So this is a picture I took in Nigeria 2 years ago, and I like it because I think it describes quite well the chaos that's happening in that little suburb. So if we have problems managing COVID-19 in the United States of America, which is wealthy, which has a lot of industries, which has the scientific know-how, which has telecommunications, if we have problems here, how are you going to manage COVID-19 in this situation? And again, unfortunately, I don't have the answer, but it's very concerning. Traditionally, we've been working on tropical diseases that are often due to worms. We've been working on Loa Loa, a disease that's due to a worm that can sometimes be seen migrating through the eye. We are working on river blindness, which is due to a similar worm where the embryos migrate through the retina and lead to progressive and permanent blindness. We're working on lymphatic filariasis, where the worms can lodge in the lymphatic system, prevent the lymph from circulating properly, and that leads to these massively enlarged limbs, as well as to secondary skin infections. And in the course of our work, we've developed an ultrasensitive platform for antibody detection. And our antibody tests are up to 30 times more sensitive than similar ones made by others. So when COVID-19 appeared, for us, it was a normal thing to do to try to retrofit our platform to detect COVID-19. And here I'm giving you the latest data. It just came on Monday, 2 days ago, from an independent lab in Florida to whom we had shipped our test. And the test came back as being 97% sensitive and 100% specific. So these are really great values. Keeping in mind that the FDA requires a sensitivity of greater than 90% and a specificity of greater than 95%. So this is really very encouraging, but contrary to what we had observed before and to what I was expecting, 4 out of 30 samples took 30 to 60 minutes to show positive in the cassette, and that's too long. So essentially, right now, we're currently working on shortening the time to readout. That was an unexpected news, but that's something we have to deal with. And so our plans is that we are now working on this very actively. Then we will initiate a clinical trial in San Diego. So we have obtained approval from an institutional review board, which is essentially an independent committee that certifies that the clinical protocol is being conducted in an ethical manner. And after that, we'll submit data to the FDA for emergency use authorization. And we plan on selling the test at $10 domestically, where each test sold would then sponsor the donation of another test to a person in need that can be in a low or middle income country, but that can also be domestically. And if we're going to a low to middle income countries, we don't have to necessarily think Africa. For instance, just across the border from San Diego, in Mexico, the situation is really bad. As for these low and middle income countries, we also need to start distributing the test as fast as we can, maybe even for free, so that the sensitivity and specificity can be assessed locally. Other countries may have different histories of non-COVID-19 coronaviruses, and that may affect the specificity of the test. So it's important to check that. And then we hope that the antibody testing will support public health decisions. We also hope that antibody tests may find some uses for individual case management and for contact tracing, even if they are suboptimal compared to a molecular test. Another way of saying that is that antibody tests may help better manage precious resources, such as the molecular tests that are already hard to get by in the United States, but are available in even more limited amounts in other countries. And then, I don't know if we're going to get there or not, but certainly our hope is to be able to come up with a salivary test for use at home and in the field. These tests should ideally detect both antibodies and antigens on the same test strip. This is extremely challenging. It's not an easy target, but it's also extremely needed. And this brings me pretty much to the end of the presentation. Before I conclude, I just wanted to let you know that DDTD is looking for volunteers for the upcoming trial. So we're seeking primarily people who've had COVID-19 and can provide a proof of a positive test, ideally by PCR. People without a history of COVID-19 may also be of interest as negative controls. if you sign up as a negative controls, you need to be aware of the fact that we might draw some blood in the unlikely event that you give a false positive result. The participants will be compensated for their times. You must be at least 18 years of age, and you must be able to come to our San Diego office. If you're interested in supporting our clinical trials, please do contact us. Contact Melissa Chalmers at melissa.chalmers@ddt.org. And if this is too long to be jotted down, you can just write to admin@dddtd.org, and we will see your email. That's it for today. I thank you for your attention, and I will try to answer the questions the best I can.
Thanks, Marco. Just to note, we will be sending out a copy of the recording of the presentation. And for those of you who would like a copy of the deck as well, you can contact me directly and I'm happy to send that. So the first question that we have is, can you explain what sensitivity and specificity mean?
Yes, so say that you have 100 people who you know have had COVID-19. Now, if you use your test, if we use our test, for instance, out of these 100 people, how many will show up positive in our test? So that's the sensitivity. If we detect 90% of them, the test is 90% sensitive. The specificity is almost the opposite. If you've had 100 people who you're sure never had COVID-19, is there a risk of false positives? So if 5% of the people who never had COVID-19 end up giving a positive result, then the test is 95% specific. And that can happen because, as you know, COVID-19 is not the only coronavirus around, there's many others.
Great, thanks, Marco. The next question is, how long after infection would you expect the antibody test to be positive?
So I sort of provided the data, right? So 3 weeks after the initial symptoms, the test should be positive. At least 90% of the people have developed antibodies after the first 3 weeks.
Great. Next question, have we sorted out which tests are reliable and have expected sensitivity and specificity?
That's a great question. I'm not sure I have a great answer, but I will show this slide of resources, and I'm very happy to share that. So there are comparison of molecular tests that have been published, and there's also comparison of serological tests, including by the FDA. So that's probably the best way of comparing tests across the board. There still remains the problem that it's difficult to compare tests if you have different types of samples, right? So the FDA may have a certain sample set and I may have another one. Would it behave the same? I don't know, perhaps not. So, but to answer the question, essentially, which tests are expected to be reliable, I would go to, for molecular tests, pretty much anyone. For serological tests, I would go to the FDA.gov website, and this link, and you will be able to at least compare data.
Great.
Maybe I'll add one more thing. If it's run on a clinical analyzer, and if it's manufactured by a big company, it's probably as good as it gets.
Okay. Next question. Will any test now or in the future be able to determine immunity to COVID?
Yeah, so this is a great question. So if any test now or in the future will be able to determine immunity to COVID. Well, I've shown a lot of the caveats about trying to predict immunity. You can still try to stack the odds in your favor. So you can try to make an antibody test that will, where was it? You can try to make an antibody test, and that's what we're doing, by the way, that will only recognize the antibodies that bind to that receptor binding domain on the spike protein. That should be pretty predictive of immunity at the time of testing, but one needs to have more data to demonstrate that. And also, I would think that we've seen that the immune response wanes with time. So on day one, you may have a strong immune response or a lot of antibodies. That may be a very good predictor of immunity. But 2 weeks later, you may need to, or sorry, 2 months later, you may need to run the test again to make sure that you still have high antibodies levels. So I'm sorry, I know it's a bit of a wishy-washy answer. You cannot predict immunity for sure, but I think we should be able to at least have data that is strongly suggestive of immunity. Sorry, I can't phrase that better than that.
Okay, great. Thanks, Marco. Next question. Is the new antibody test reliable enough that people who test positive don't have to quarantine?
Well, I don't know if the question relates to our test or antibody tests in general, but regardless, no, because as we've seen, people can develop antibodies slowly. After the first week, only 30% of the people have antibodies. After 2 weeks, we're maybe in the 80% range. After 3 weeks, we're in the 90% range. So So by the time you have antibodies, you probably have already gone over your quarantine time.
Okay, great. Thank you. Next question, and then we've got a couple more. How realistic is it to expect that we will be able to order a packet of 50 to 100 tests and do in-home testing on ourselves every few days?
Yeah, I love that question. Again, I wish I had the answer. I think it's not going to happen anytime soon. I think we're at least 1 year away from that happening for a number of reasons. One of them is just the technical difficulty. You need to have a test that can work super well and just be read with the naked eye or with a very cheap instrument. And that is already difficult in itself. In addition to that, we've mentioned the issue with sampling, and sampling is actually not easy, right? You need to come up with an alternative to these nasopharyngeal swabs. So that's another difficulty. And the third difficulty is that the FDA, and rightly so, will require vast clinical trials. So you need to show that thousands of people, regardless of their skill level and understanding of the English language and what's not, will be able to perform the test accurately. And that just demonstrating that will require months. So I think we're at least 1 year away from that. I hope I can be proven wrong.
I think we all do. So I have just a couple more questions. What is the status of using pooled testing as a testing option? We would consider this for our employee group if this could be a reality. As I understand, it could lower testing costs significantly.
Right, so the question is about pooling samples, and the question is, okay, these molecular tests are expensive after all. Can I take swabs from 4 of my different employees, mix them together, and have only one test run on the mixture of the 4? I've not followed the latest news in this respect, but I think it's a perfectly feasible option. I think it's a smart option. And the truth is that the disease prevalence, though, how can I say, though the news have been dramatic, is still very low, right? There's maybe 1 or 2% of people, at least in San Diego, who've had the disease. So the chances that you inadvertently mix a positive sample with 3 negative samples are very small. So I think it makes a lot of sense to mix 4 samples and send them. It cut your cost by, a quarter, at least on the testing side, maybe not on the sampling side. And then if you have a positive, then it's easy to go back to these 4 individuals. You do need, however, a fast turnaround time for that to be useful.
Great, thank you, Marco. Second to last question. How can we protect our loved ones who live in a senior care facility?
Yeah, that's a lovely question. From I don't know how to answer that one either, but and maybe it's going to be slightly beside the point. So yes, of course, you can hope that the people in the senior care facility get tested, both the personnel and the people, the seniors in the facility on a fairly frequent basis with a molecular test. There's another thing that I think makes a lot of sense, and it's not a very expensive option, and it's to retrofit the airways, essentially the ducts, with system that can purify the air against viral particles. So you can have filters installed, but the filters aren't that great because they can block bacteria, but the viruses are often too small for filters. However, you can combine that with UV light, and that's the system that's used in hospitals. So I know that I've done that at our house for $1,300. We're going to do that for our lab here so that our personnel is protected. I think that is a pretty cheap way of protecting the people you care about.
I agree. Thanks, Marco. And for the final question, how will DDTD's antibody test results feed back into the bigger picture to help health organizations better understand the virus?
Wow, that's the hardest question for the end, right? Well, there's two things. One thing that I've not maybe been telling very well is that we are about trying to deliver high quality at low cost. And we're really pushing hard to make sure that our tests are reliable. So at least that's one thing we're trying to offer. Then how the data feeds back into other organizations, well, again, I think it's about understanding which pockets of a country need to take more precautions than others. I'll try to maybe answer that question with something that's close to my heart. So there's some people in Ghana, Africa, with whom I'm still connected. I visited the place some 20 plus years ago, and they still call me every now and then. And they're very modest people. They live in a fisherman village. The fish is scarce. They have very few resources. They're confined. It's very, very, very hard for them. But there's also no sign that there's been COVID-19 in that village. So if you could prove with an antibody test that that village is COVID-free, then you could at least release those people and allow them to go back to their tasks. Simple as they may be, they can go back fishing together or working the field together. And I think if we manage to do that, that would be a plus.
Great. I did actually have one last question, Marco, and then we'll wrap. How much help is daily temperature taking?
So I didn't hear the question. How much?
How much help is taking your temperature daily?
Yeah, I don't know. I think I'll pass on this question, right? It's known that maybe anywhere from 20 to 40% of the people can be asymptomatic. So I think you'll still miss a number of asymptomatic people. But if you can catch the few that are symptomatic, and if you manage to reduce the transmission, would it only be by 50%, that can make the difference between a country or a city that can manage COVID-19 and one in which the healthcare system is completely overloaded. So all that can be done to reduce transmission, even if it's just by a little, I think it's worth it. I also think that most people will know if they have a fever without necessarily measuring their temperature.
Mm-hmm. All right, well, thank you to everyone for joining us today for understanding COVID-19 testing. Again, if you'd like a copy of the deck, you can email me at melissa.chalmers@ddtd.org or admin@ddtd.org. We will be sending out a recording of the presentation within the next day or so. Thank you again for joining us, and we hope you found this informative.
Thank you. Have a beautiful day, stay safe, and I hope that I'll see you face to face one day. Bye-bye now.